Hi, I'm Jim Heath. I'm the president of the Institute for Systems Biology, and I'm here with my student who recently departed with a Marshall Fellowship and is now at Churchill College in Cambridge in the UK. And first, Daniel, congratulations to both of us on getting this paper accepted to Cell Reports. So this is a paper, Daniel Chen is the first author. The title is a mouthful, but it's actually a pretty simple story. The title is T-cell receptor sequences are the dominant factor contributing to the phenotype of CD8 plus T cells with specificities against immunogenic viral antigens. So once you get past that mouthful, it's actually a pretty straightforward story. So Daniel, why don't you give us a rundown of what you found?
Sure thing. The basic idea of what we found is that a T cell that usually, attacks cancers and virus-infected cells, a lot of questions have been raised about how is this phenotype determined? How do we decide, is this T cell going to be really effective? Is it going to be more memory biased? Will it survive? And what we found here is that the receptor sequence, basically the protein that's on the T cell that binds antigens, that binds these foreign antigens, that receptor sequence is the major determinant, not cytokines, not what's in the plasma, not any other factor. It is that receptor sequence that determines what this cell's phenotype is, and thus also determines what its function is going to be. And that's a story.
I think it's striking. This simple observation was, it goes to it's as fundamental of a biological observation as one can imagine. And that just says that the function that a T cell has after it's been activated by viral antigen is completely dictated by the T cell receptor genes. Most often in biology, in studies like this, one designs a model system, such as a mouse model or something like that, to really do detailed tracking of some T cell and figure out what it does over time. But that is really not what you did here. What you did is actually much more relevant to human disease. And so why don't you describe the basic method by which you found this?
Exactly as you said, we're looking at real human patients with real clinical disease. So we've looked at hundreds of COVID-19 patients, and we filtered down to those that we really could study. So a model system in a real clinical setting, those with a specific genetic genotype. So those that we can actually measure and know for sure their T cells are specific for either SARS-CoV-2 or other viruses. And what I think was really interesting that we did here is we not only said, what are the cells specific for the active infection, but what are also cells that are specific for infections that the patient does not currently have? So not only are active infection cells, their phenotype are determined by their receptor genotype, but it's also all the other cells, those from like the flu that, happens every single year, those from other latent infections like CMV. Even if there are other factors that might be involved to certain degrees, there's been a lot of reports about how some T cells can be activated just from cytokines. It's still that receptor gene that is the most important, even when all these other factors, even when real disease is prevalent. It's that receptor determined way back in the thymus in early childhood that really determines what that cell becomes.
Yeah, that's pretty cool. And the fact that you saw that this was true in not just in a real disease setting, but also you saw it was true for flu-specific T cells and cytomegalovirus-specific T cells and SARS-CoV-2-specific T cells. For the examples you looked at, did you find any counterexamples where it wasn't true?
We did not.
Yeah, it argues that maybe this is actually a general rule. And what's striking about such a general rule is that it begins giving you some engineering principles. If you were thinking about how would you use this as a set of a general rule set for, say, engineering a patient immune response against some disease, how might you use it?
Sure. I think one example is right now in clinic, there's this huge push to make the most cytotoxic, the most effector T cells that we can. But there's this also huge problem that comes with that, in which we infuse them into the patient and they burn out. We don't see them after a few weeks. And so there's this need to basically go along this axis of memory, and effector, and be able to provide patients with both of these cells that can survive a long time inside of the tumor, but also cells that can go and function and have this strong effector function for that initial push. And having and knowing that we can have both of these at the same time, we don't have to just choose one, and have this combined response and combined infusion product that will help patients truly recover, not just in the short term, but in the long term.
Yeah, I think that's right. I think especially as you begin thinking about the place where engineered immunity is most important right now is in cancer. When you are engineering that immunity now, it's really with blunt tools. And what this suggests is that there's basically a card catalog available, and you want a particular prescription of an immune response, you go to that card catalog and you find this particular TCR genotype or one that closely matches it, because you found that genetically similar T cells actually had genetically similar functions. So it wasn't just a random assignment from one gene to another. It actually sort of made sense. So I think that that's actually pretty exciting. So what's next? What do you think would be, I guess, maybe showing that this type of engineering could possibly work?
Yeah, it would be really exciting. I think there's so much data that's already out there, and there's so many neoantigens that we can definitely create these personalized databases of what are the patient's T cells, what are the receptors, and if we know, as you said, there's this relationship between as genotype goes from like point A to point B, so does phenotype, then can we take a given patient's genotype and take the given patient's phenotype, take samples that we want, as you said, like take a catalog from these different points, and then provide them with that personalized therapy.
Yeah. And I think also something that's, worth mentioning here that probably wasn't so obvious is that when you saw that a bunch of genetically similar, not identical, but genetically similar T cells all had the same functional phenotype, those genetically similar T cells were coming from 60 or 70 different patients.
Right.
And so that does suggest that, what you learned here is actually quite general across patients. Even if we, for a given immunogenic antigen, still have to work out what are the specific genetic rule sets for that antigen, they should hold firm.
Exactly. I think that's a really important point in which we did find these general patterns, and they are personalized for the antigen, but they are shared across these different patients. So likely what we can identify for one patient is going to be applicable for another. I think that's a nice generality.